Hi, my name is Arturo Ramirez, and I'm Director of Oncology Research and Development at RareCyte. In this presentation, I will go over RareCyte's technology for comprehensive phenotypic and genomic characterization of CTCs. RareCyte is a precision biology company that provides instruments, reagents, consumables, and services for deep CTC characterization. The key features of our platform for CTC analysis are outlined here. Our technology is an end-to-end solution for CTC characterization. Blood is collected into vacuum tubes with a proprietary preservative that provides a 3-day blood stability window. At a processing lab, nucleated cells are adhered to microscope slides, stained with our RarePlex staining kits, and scanned on a SiteFinder instrument that includes integrated machine learning algorithms that facilitate concordant CTC identification. Once cells are processed to slides can be banked for over a year prior to staining, allowing sample collection to proceed ahead of assay development. Furthermore, high-quality plasma is obtained during sample processing, enabling cell-free DNA analysis in addition to CTC analysis for a more comprehensive liquid biopsy test. RareCyte offers biomarker developer technology to add custom biomarkers to CTC enumeration assays with 2 available open channels. We use this technology for assay services and train customers how to use it to add markers of interest in their own labs. Our RarePlex assays for CTC enumeration and biomarker expression analysis are designed to pharma and CLIA standards, with an emphasis on accuracy, precision, and reproducibility using quantitative imaging-based decision-making during assay development and validation. Here are examples of clinical CTCs found with our assays, showing our standard approach to detection of epithelial CTCs as nucleated events with CK or EpCAM expression that lack CD45. We also have 2 open channels for phenotypic characterization of CTCs. The high sensitivity of our assay results from the way we identify CTCs. We don't perform CTC enrichment based on size or rely on particular surface marker expression to capture cells, but rather we employ high-definition fluorescence imaging and machine learning algorithms to perform the identification of cells of interest. To quantify CTC enumeration accuracy using our platform, we first tested spike-in surrogate samples. The chart on the left shows a typical result from one of our validation studies for enumerating spike-in cells. We deposited 5 to 8 culture model CTCs into blood samples, processed in a blinded fashion using our epithelial CTC enumeration assay, and identified 47 out of the 49 model CTCs across 9 replicates for a 96% recovery. To assess clinical accuracy, one of our customer labs performed a blinded comparison study as part of their CLIA validation of the RareCyte assay, with results shown in the plot at right. Sequential blood sample draws were obtained from 50 prostate and breast cancer patients and processed using cell search and RareCyte assays to enumerate CTCs. The correlation and slope between the 2 assays indicate very similar performance, with RareCyte identifying on average 30% more CTCs per sample. Taken together with spike-in results, this data highlights the accuracy of the RareCyte assay for detecting CTCs. To answer the question of how clinically useful CTC count is with RareCyte technology, this slide indicates, not surprisingly, that CTC number at baseline is an independent predictor of progression-free survival, even in a small and heterogeneous population of solid tumors from a clinical study of participants undergoing checkpoint inhibitor therapies. A key highlight of our platform is flexibility to meet the user's research goal. One example is the availability of 2 extra channels that can be added to the CTC detection assay. Those extra channels provide expression and localization data on the identified CTCs. Our developer kits allow any lab to use mouse and rabbit antibodies to stain for extra biomarkers, and this technology is the same one we use in our lab to design new assays. As an example, I'm showing a lung cancer assay that has Vimentin and EGFR as extra markers. A second example of our flexibility is the cell discovery kit, which allows the substitution of the human CD45 for mouse CD45 to perform rare cell detection in mouse blood, and allows the substitution of CK and EpCAM for another cell detection marker to allow for non-epithelial rare cell identification. As an example, I'm showing an assay made to detect melanoma cells by adding the S100 protein to the nuclear and CD45 channels. Here is a non-comprehensive list of biomarkers we have worked on. Due to client confidentiality, most of the markers we have worked on are not included. We also offer assay services to leverage our extensive experience delivering custom assays for both pharma and academic partners, with an emphasis on accuracy, precision, reproducibility, and clinical data, all wrapped into an end-to-end CTC platform. Biomarker accuracy is an important metric that we quantify during assay development. When validating a new assay, we select positive and negative cell lines, set the threshold that best separates the two populations, then measure sensitivity, specificity, and accuracy of the assay. Single-cell mean fluorescence intensity is automatically generated, and the determination of biomarker positivity is a yes or no answer that is user-independent. In addition to biomarker status, the software outputs single-cell mean fluorescence intensities for each cell to quantitatively measure expression in addition to biomarker, localization, and morphology, which in some cases can be as important as signal intensity when characterizing protein expression. Single-cell genomic analysis is made possible through the Site Picker module installed in some models of our SiteFinder instruments. The workflow for isolating single cells is highly automated and imaged in real time as it occurs. It uses a microneedle to physically detach the cell from the slide and deposit it into a proprietary PCR tube for visual confirmation. Now the cell of interest is ready for downstream molecular analysis. Each cell takes about 3 minutes to pick and dispense. We and our customers have performed a multitude of clinical genomic characterization studies, and due to time constraints I can't go into too much detail here, but what I will say is that our technology has been used to perform studies that range from Sanger sequencing to look for single nucleotide polymorphisms, targeted sequencing on a few dozen common cancer genes, copy number variation, all the way to whole exome and whole genome sequencing on single CTCs to characterize tumor evolution over time. Our results have recapitulated known mutations from tissue sequencing data, but allow us to probe tumor evolution over time that is not possible with tissue biopsies. We have used the high-purity plasma that is collected as part of our protocols and compared genomic information between CTCs and cell-free DNA and found that CTCs and cell-free DNA are complementary sources of molecular information, with some variants found only on CTCs and others only in the plasma, while most are found in both compartments. Our philosophy is that the information contained in CTCs has added significance as it represents the tumor compartment which holds information about the metastatic potential of the disease and may represent a resistant phenotype, as opposed to the cell-free DNA compartment, which may be used for early detection or minimal residual disease monitoring, but is generally thought of as the DNA of cells that have died and therefore no longer have metastatic potential. Also, CTCs are better sources of DNA for de novo mutation identification as they are pure genomes, as opposed to cell-free DNA, which is mostly non-malignant in nature. In conclusion, I will end by highlighting that RareCyte has a variety of products and services for liquid biopsy available, spanning anywhere from discovery research to clinical trials. Our key differentiators are the ability to develop multiplex assays, sequencing single cells, and an end-to-end solution for liquid biopsy evaluation, all on a microscope slide-based platform. Our comprehensive, flexible, and reproducible technology is available to assist you in your research goals. Thank you very much for your attention.